Okay, so we're going to talk right now about DNA duplication. And so when we duplicate DNA, what we mean is that you've got a chromosome, and remember that your chromosome is, if we're going to duplicate it, it wouldn't look like that. It's going to look like this. And we need to duplicate the information that is contained on this side of the chromosome, which means we're going to go from something that looks like a stripe, and when we're done, it should look like an X, because we've got a sister chromatid over here that is an exact copy of everything that you see on this side. And so this piece of DNA, if we zoom in further, remember it's not really like a line, like a stripe like that. It's actually two strands of DNA that are making this double helix structure. And so this is equivalent to this. Those are the same thing. This will just be a higher magnification view of the DNA that you would see over there in that chromosome. So we need to go from one copy of the DNA to two, and every single piece of DNA that is on these two strands needs to end up in the other one. And so the kind of duplication that we are going to do is something called semi-conservative replication. And that is because when we make our new DNA, it's going to have half of the old information, and then the other side of this double-stranded molecule will be new information. So the chromosome's really, really big, and because it's so big, we're not just going to start duplicating in one area. If we started the process in only one area and then did the entire chromosome, it would take a really, really long time. So if we zoom in on a strand of DNA, instead of it being just one spot where it's being duplicated, the DNA is going to look something like this. There's going to be a whole bunch of different areas where duplication is occurring. In each one of these areas where you see it swelled outwards like this, we call this a replication bubble. And then these parts kind of right here within that replication bubble, kind of like right there or right there, you usually call those a replication fork, because it's got this Y sort of a shape, kind of looks like a fork, and those are the boundaries of the overall replication bubble. So if we zoom in on this area of duplication, in my drawing that I'm going to make, I'm only going to really be drawing like one part of, like one of these bubbles, not the entire thing. So what I'm drawing is going to be happening on a whole bunch of different locations within the DNA. So if black represents our old piece of DNA, it's going to be opened like this. And so we got our two sides of the DNA molecule. And remember that those base pairs are bound together with hydrogen bonds in between all of those different base pairs. And so that's what we see over here. These indicate bound nucleotides. And then this is an area where they've been opened up. So in the areas that have been opened up, we're going to have exposed bases. And so I'm just going to put some bases in here. I'm going to try to make sure that I've got equal numbers of bases on either side. And just to add to our drawing, I'm going to go ahead and give these identities. So we'll say it's like C, C, G, T, A, C, G, just to give it some bases. And you should be able to name what's on this side. So if that's a C, you know that this is a G, G, C, A, T, G, C, because they're complementary of one another. And so the way that we went from these bound nucleotides to these exposed nucleotides is through the action of two enzymes. And these are the first two enzymes we're going to talk about. And these enzymes are responsible for opening up the piece of the DNA and separating these nitrogenous bases, which are bound by hydrogen bonds up here in this picture. And so those two enzymes that would be out here, there's going to be one right here kind of closer, and then one that's over here a little further out. And in this picture that I'm going to draw, I'm going to say that they are moving in this direction. Okay? And so those two enzymes are called Helicase and Topo Isomerase. So helicase is the one that is always closer to the bubble. And helicase's job is to open up this double-stranded molecule. It's to kind of unzip the DNA. And so it's going to come onto the DNA, and it's going to start piecing it apart like this. And in my picture, it's moving from left to right. Whenever that happens, though, because you started out with this double-stranded molecule, and you're kind of pulling it open in the middle, you can imagine that you have like a strand of string, and the string has got these fibers that are wound around one another. Well, if you take that string and you grab it in the middle and just pull it open like this, you're going to create a whole bunch of stress on either end of that string outside of the area that you're opening. Because as you open it in the middle, you're winding all these other areas on the side more tightly. And so that's exactly what's going to happen over here, is that as you open up the DNA, you're putting a lot of stress on these outside regions where you're causing it to wind up even tighter than it was before. And so the enzyme that you find further to the outside is Topo Isomerase, and it is there to relieve tension on the DNA. As helicase opens up the DNA and it winds all these pieces tighter, Topo Isomerase is going to cut the DNA, allow it to spin out, and then glue it back together again. And so helicase has a job of opening the double helix, whereas Topo Isomerase has a job of relieving tension on the DNA. So those are the two enzymes that are involved with opening this thing up. But after they get open, you can imagine it like these are magnets on either side. These are all North Pole magnets, and these are all South Pole magnets. Well, as soon as you pull them apart, all they want to do is slide right back together again. And so we got to put something on there to keep them open. And so bound to every single one of these nitrogenous bases is going to be a little bitty protein that just kind of sticks to that nitrogenous base so that way it kind of caps it, and they don't want to slide back together again. And all of these little proteins that keep it from snapping back together, these are called single-stranded binding proteins. And they're usually abbreviated as SSBPs. And so SSBPs bind to nitrogenous bases to keep the piece of the DNA that's been opened up open so that we can actually bring in the next set of enzymes that are going to work with this piece of DNA. And so this is kind of the first round. This first round of enzymes are helicase, topoisomerase, and SSBPs. Their job is to open up the DNA, relieve the tension, and then keep the bases separated so they don't go back to the way they were before we began. And so those are our first three. The next step is now we got to actually start making new DNA. And when we make new DNA, the enzyme that we want to eventually bring in is one that is called DNA polymerase. But DNA polymerase is what makes new DNA. And DNA polymerase isn't really going to do its job until it has kind of had the whole piece of DNA kind of prepped for it. And the way that we're going to prep for it, I'm going to kind of draw another picture of this next step down here just so we don't end up adding too many things to the same picture. We want to be able to see what we're doing here. So these are our bound guys over here, and we had 1, 2, 3, 4, 5, 6, 7. So 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7, and it was C, G, T, A, C, G, 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 C, A, T, G, C. So we've got all of these guys down here, and these are the same ones that we drew before. So now that we have it open, the next step that's going to happen is another enzyme is going to come in. And this enzyme is named primase. Primase is going to lay down something called a primer. Okay? So I'm going to draw primase after it's already actually worked with a few of these bases. And primase comes in, and it lays down this primer. And this primer is a piece of RNA. And this piece of RNA is going to be the complement to the template strand. And the bases on here are just going to be what you would normally find on the other side. So C binds with G, C binds with G, G binds with C. And so this piece of RNA, which we've laid down, that is called the primer. And again, it is made from RNA. The enzyme that has laid down the primer is called primase. And in my picture, it would be moving to the right. And the way that I know it'd be moving to the right is because it's only going to lay down RNA in the places it's actually gone. And so if this is where you're going to find the primer, that means the primase must have already been in that position. And so it's moving in this direction. And just to add into my picture what I had before, here's my helicase, and then here is my Topo Isomerase. And so primase is moving along, it's laying down this primer, and this is basically the new information, the new pieces of DNA that are being bound to that template strand. Now that we got a primer in position, we can now bring in the next molecule, which is the most important one, which is the DNA polymerase. And so DNA polymerase is going to come in, and just like before, I'm going to draw a new picture just so that we don't have too much stuff jumbling up what it is we're trying to do. We got 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7. And we would have already laid down our primer. So here's my little primer down here. And it was G, G, C. This guy was C, C, G, T, A, C, G. And then we got G, G, C, A, T, G, C. Okay, so we've got our primer right there, so I'll go ahead and label that. Here's our primer. And now we've got our DNA polymerase. So DNA polymerase is going to be kind of in this section. I'll draw it after it's already interacted with a few of these bases. And so our DNA polymerase is right here. And it has, in this picture, it is also moving towards the right. So if it's moving towards the right, that means the bases it's interacted with are these right here, because that's where it was before it was in this position. And so the DNA polymerase is going to lay down new DNA. So here's my new DNA. And it's going to have the bases that are complementary to the template strand. So this would be an A, this would be a T. And so we have this piece of primer, which is RNA, attached to new DNA, which is going to be complementary to the template. The primer was laid down by primase. This new DNA was laid down by DNA polymerase. And in this picture, you can see that it's moving to the right. And so our helicase, just like in our other pictures, would be right there. And our Topo Isomerase would be right there. And so in this picture, everything moving left to right, just like we had it in the very first picture that we drew. Okay, so everything here is fine. We've got our initial strand that was unwound, unzipped. We got a primase that bound DNA polymerase that started making new DNA, and now everything would just keep moving in this picture from the left to the right. So now if we think about this in the larger context of the entire chromosome, and go back to kind of the first thing we were drawing, if we're looking at the chromosome as a whole, this process is happening millions of times throughout the entire chromosome. And they're all going to start kind of at different times. These enzymes come in and attach to the DNA, but they don't all start at exactly the same time, which means that some of these bubbles, you know, they've been going on for a really long time, and so they might be really big. And then some areas may have just began, and so we've got a tiny little bubble. And so if we're to look at these bubbles, we'd have like areas where you've got your RNA primer that was laid down, and then that is attached to new DNA that's being produced. So in this particular drawing, I'll have this be equivalent to the primer, and then this will be equivalent to new DNA. And I'm going to keep drawing that, and I guess the thing I want you to notice is how these are not always going to be the same. So look at the location. Oh my gosh, I just messed that up. Let's pretend that isn't there. Okay, so look at the location of these things. What I want you to notice is that they're always opposite. If the primer is on the left in this particular bubble, it is on the right on this one. If the primer is on the left over here, then it is on the right, then it is on the left in this one. So they're always kind of in an opposite orientation. And also notice that if I'm talking about this top strand, in this particular bubble, the primer is on the left, the new DNA is on the right. But in this one, the primer is on the right, and the new DNA is on the left. So it is true that these guys are always opposite of one another, but whatever is happening here doesn't have an effect on what's happening over here. Okay? And so you could potentially have two that totally look the same right next to one another, like these two, for example. This one could have been there, or this one could have been there, could have been alternating or the same in a row. That part doesn't matter. But this one is always opposite relative to that one. Okay? And so the other thing you should be able to look at and kind of figure out is that if I'm looking at this particular bubble and I ask you, where is the DNA polymerase? Well, you should be able to know that the DNA polymerase in this picture is in this location. And the reason you know that is that the DNA polymerase is always going to be laying down new DNA behind it. The new DNA is always behind the DNA polymerase. It's like an exhaust trail or like tire marks or something on the street. they're not going to be in front of the car, they're going to be behind it. In this particular bubble, the DNA polymerase is at this position. In this one, it's at this position. And this one, it is at this position. And then if I go to the bottom, it's the opposite. And that little error right there, remember that little red isn't supposed to be there. And not only do you know where it is, you also know which way it's moving. So here, the DNA polymerase is moving there. Here, it's moving there. It's moving there, here, there, 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 and there. And again, it's because it always has the DNA it's made is behind it, not in front of it. And so these bubbles are just going to keep on going in whatever direction they are, and eventually they're going to counter one another. And so if we were to extend this thing out and look at this thing over time, this bubble is going to expand and probably eventually reach that one. And then these two are going to expand and they're going to reach one another. And when they encounter one another, you're going to have areas of primer right next to areas of new DNA. Well, primer is made from RNA. RNA is not meant to be a long-term sort of a molecule. It's short-term, it breaks down over time, it needs to be replaced. And so when this one reaches that one, you're going to have a stretch of primer over here, and then you're going to have your new DNA. When that DNA polymerase encounters this DNA polymerase, they'll just kind of merge together. And now you're going to end up with primer on both ends of this particular molecule. Over here, you're going to get primer that's over here on the left. And then when this DNA polymerase encounters that primer, like when this meets there, it will remove the primer and replace it with new DNA. And so that means that you're going to end up with just a solid piece of DNA going along here because that primer will be replaced. And then that's going to continue as this DNA polymerase keeps moving over there to the right. And so here, they're just going to fall off. Here, it's going to keep moving over to the right. In this particular one, when they join together, you're going to have your primer over here on the left. This guy's going to encounter that one. It's going to knock it off. And you're going to end up with something that looks like this. Here's your polymerase moving across. When these guys encounter one another, I take that back. They're actually going to end up with the primer kind of in the middle on this bottom end. And then you're going to have your polymerases moving in both directions. And you can see how what's eventually going to happen is that all of these DNA polymerases are going to meet up. All these primers are eventually going to be replaced, and you're going to end up with your new strand of DNA. To where in the end, you've got both template strands right here, and you're eventually going to have your new DNA extending all the way across. Okay? And when you get down here, this will be your final product, and DNA duplication is going to be over. There is a problem that is going to happen here that will arise, though. And the reason that you're going to have a problem is because you'll notice that every time that you duplicate the DNA, you're always going to have a little bit of primer that will be on the ends that is never going to be replaced. And that's because since it's all the way over here on the end, like not this particular primer, imagine there's a piece all the way over here. There's nobody else that's over here to like the left who's ever going to encounter this primer. And over here, there's never anybody over here to the right that will encounter the primer that's laid down on the very tip. So you're going to always end up with a little piece of primer down here, which will not be replaced, and that's going to be a problem. And that's what we're going to talk about in class. This is going to lead to something called telomeres. So this is kind of the overall context of what's happening within each of those little replication forks, is that they're all expanding at the same time. Whenever they encounter one another, which is going to happen pretty regularly, you just overwrite the primer, and you replace it with more DNA. And eventually, once they all meet with one another, you're going to end up with a continuous piece of DNA going all the way across both sides of the molecule, and that's how you get these new strands of DNA. And so if this kept going, these two, they're going to be one piece of DNA, and these two, they're going to be one piece of DNA. And so when they actually do their double helix, you're going to have them joining together to make the mature structure like this. And the two top are going to become this top piece of DNA, and then these two on the bottom are going to become this one. I hope what you're noticing is that we're leaving out some information, and that is because I described to you in this picture how we are going to start producing new DNA along this top strand after the primase has done its job of laying down a primer, and after the DNA polymerase has come in and started laying down this complementary strand. And we drew this picture over here. This is the new DNA that's being made by the DNA polymerase. And so this is pretty much equivalent to this. Sorry, make sure I'm showing this. this is pretty much equivalent to this. This is just kind of a zoomed out view of what's going on in this picture. Well, what I never talked about is how do you get this other side? Because you might not have noticed it, or maybe did, but this other side is going to have a problem. And the problem is that in this particular image, everything is moving from left to right. The helicase is going from left to right, Topo Isomerase is going left to right, which means that this little bubble is opening up in this direction. This side is always getting bigger, but nothing's happening over here on this side. And so this side is not expanding. And so when we start laying down new DNA, if you're on the top strand, everything's okay, because you can just keep on laying new DNA. Your helicase and Topo Isomerase are leading the way for you, and you're just going to keep on going. And in this image, that's what's going to happen. You're just going to keep on going in this direction. Well, if you're on the other side, if you're along this bottom strand right here, you have a problem, because you want to start making DNA, but DNA has something called a polarity, which means that DNA has like an up, down, left, right. DNA is not just universally the same in all directions. DNA has a polarity, and the two sides are called 3' and 5'. And what that refers to is the number carbon on this sugar. They all have a pentose sugar, deoxyribose, that's sugar. Whichever number carbon is sticking out on the bottom. And all of these enzymes read DNA 3' to 5'. So the helicase is reading the template 3' to 5'. The primase is reading the template 3' to 5', and the polymerase is reading it 3' to 5'. So that means that in all these pictures, this side is 3' and this side is 5', because in all of these pictures, they're moving from the left to the right. So that means that the 3' must be on the left, and the 5' must be on the right. Well, the polarity of DNA is not parallel, it is anti-parallel. And what that looks like is these two markers, right here I'm holding them in a parallel orientation. But if I want to hold them in anti-parallel, it would look like this, meaning that they're both still parallel in terms of their overall relationship to one another, but they're going in opposite directions. And so it's anti-parallel. They're still parallel, but they're not facing the same direction. And so if this is 3', that means that this side has to be 5'. And if this is 5', this has to be 3'. So when I fill that in, you're going to see what the problem is with this whole setup. The problem is that all of these enzymes are moving 3' to 5', which means they're going to be going like left to right in this picture. But the enzymes that are going to attach to the bottom strand, the same ones, DNA polymerase and primase, once they attach, in this picture we had primase coming up here, that means primase is going to attach right here, and it's going to make the primer over here on this side. So A, C, G. But this primase is moving, instead of left to right, it's going right to left. And when the DNA polymerase comes in and does its job, it's going to be making new DNA on this bottom strand. So here's a T, here's a G, and then here's our DNA polymerase, but it's going in this direction. And so that means that this enzyme wants to move to the left, but the little bubble is only expanding to the right. And so this enzyme is going to hit the wall every time it gets started. It's going to get started, it's going to start making DNA, and then it's going to go boom and bump into the edge of this bubble. In these pictures, it'd be like, imagine that you are this enzyme, you're moving over here to this direction, but this bubble is only expanding that way. It is not expanding this way. And in this picture down here, you see what's going on there. This bubble is expanding out here, but not in this direction. And so how do you start duplicating the DNA that's over here, or over there, if your enzyme is heading the wrong way? Notice on this side, it's also heading the wrong way. And so what is going to happen is you're going to make these things that are called Okazaki fragments. Gotta get a new piece of paper. Here we go. Okay. You're going to make these things that are called Okazaki fragments. And Okazaki fragments are the way that you're going to duplicate the DNA on the bottom. And what that means is that you're going to have your piece of template DNA. It's going to look like that. And on the top, you're going to have like your primer, and then you're going to have your new DNA, and then you're going to have your DNA polymerase. And then outside of that, you got your helicase, you got your Topo Isomerase. And in this picture, they would all be moving this way. Well, your primer for your other one would be right here, and then the new DNA that you made would be going out like this far. And then you'd have another little primer. and then another piece of DNA with another DNA polymerase, and then another little primer, and then another piece of DNA with a little DNA polymerase. And all these guys are headed over here to the left. And so what's going to happen is that your strand on this side that is going the same direction as the helicase and Topo Isomerase, it's going to continually synthesize. On this side, you are going to have continuous synthesis of DNA. And because of that, we call this the leading strand. Over here, you're going to have discontinuous or uncontinuous synthesis. Of DNA. And so this is called the lagging strand. And so as this bubble moves from left to the right, this is going to keep on building new DNA. And then over here on the bottom, we're going to have a little piece, and then 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 a little piece. And we're just going to keep on making one little piece at a time. And even though we end up with all these little pieces, every time one of these polymerases encounters another primer, it's just going to gobble it up and replace it with new DNA. So that we still end up with our continuous piece of DNA by the time we get to the end. It's just that on the leading strand, it was with one long build of DNA, whereas on the lagging strand, it's from joining together multiple little pieces. These little pieces that you make along the way, these guys, these are called Okazaki Fragments. And I'm going to kind of cheat here and show you a picture from the book, just because this is a lot going on, and it's much easier to understand if we just look at the figures that are in the book. And the book does a very good job of describing these things for you. You can see in this picture, this is what they're talking about, is that you've got your leading strand right here, it's continuous synthesis, but you have all these little pieces on the opposite side, which are being made in the other direction. And the reason why you have to do this, again, is because the bubble is expanding in a direction that is opposite from your direction of synthesis. So rather than having one big piece, you've got a whole bunch of little ones that you will just glue together in order to get your mature